Hi students, good morning to everybody. We were talking about that is the composition of blood. We already completed the plasma. The second component present in blood, the formed elements. The formed elements include the red blood corpuscles or erythrocytes, the white blood corpuscles or leukocytes, the platelets or thrombocytes. All these three elements together called formed elements. Now let's take the first one, the red blood cells. As you already studied in your 10th standard, so it's very easy to understand that one. Now if you are comparing the number of cells of Rbcs, Wbcs and platelets, now the Rbcs are more abundant than any other types. For example, if you are taking the ratio of Wbc and Rbc, for every Wbc, there are about 700 Rbcs. This is the Wbc, this is the Rbc. So that means more abundant cells in the blood Rbcs when compared to other elements. Now the second one regarding the number. So it contains about 5 to 5.5 million of Rbcs per cubic millimeter of blood in a healthy man. The same number is not present in the case of female. If you are counting the number of cells, that is Rbcs in women, it's about 4.5 to 5 million per cubic millimeter of blood. That's a number. What about the size? The size normally very small with a diameter of 7 micrometer. 7 micrometer. Then color. The color of the Rbc is red. This is the why. This is why we can say Rbcs are red blood corpuscles. Now this is because of the presence of a red respiratory pigment called hemoglobin. We already studied about the nature of hemoglobin. Each hemoglobin molecule carries 4 molecules of oxygen, that is oxygen carrying capacity. In terms of volume, 100 ml of blood carries 20 cc or 20 ml of what is called oxygen, that is oxygen carrying capacity. So its role, transport of gases and exchange of gases between the cells and the surrounding fluid. Now we have to go for other related structures. And here is a diagram given. This is a surface view. So normally the Rbcs do not have any nucleus. The central one is not the nucleus. The outer portion, elevated one, having a diameter of 7.0 micrometer. And if you are making a cross section, the cross section is showing what we got 2.5 micrometer. That is actually the height. If you are making the cross section, this is what we get the diameter, this is the height in cross section. Okay, what is the role of hemoglobin? Hemoglobin helps the transport of oxygen particularly and facilitates the exchange of gases with the fluid outside the cell. Between the cell and the fluid outside it, nothing but the tissue fluid. This is what is called the tissue fluid. With the fluid outside the cell, nothing but the tissue fluid. The one which is present between the cells. That is called as the tissue fluid. Now, what is the importance of biconcave nature of the Rbc? Why is it biconcave? So the biconcave shaped Rbcs increases the surface area to volume ratio. Increases the surface area to volume ratio. Hence, oxygen diffuses quickly in and out of the cell. In and out of the cell. In and out of the cell, maximizing what is called the diffusion of gases, maximizing the diffusion of gases into and out of the cell. That's why the biconcave shape of Rbcs. Why is it become biconcave shape? This is to increase the surface area to volume ratio. Hence, oxygen diffuses quickly in and out of the cell. Now, if you are considering the Rbc, it is a cell, but it is without any organelles like nucleus, mitochondria, endoplasmic reticulum, ribosomes, etc. Why are these organelles absent? So the absence of these organelles normally accommodate more hemoglobin, increases the accumulation of more hemoglobin, thereby maximizing the oxygen carrying capacity of the cell. That's the reason why normally Rbcs carry just quickly and also maximizing the transport of oxygen. This is because of the absence of the organelles. So more hemoglobin is accommodated in the cell, thereby actually maximizing the oxygen carrying capacity of the cell. Okay, that's why normally we have the absence of organelles. So these two properties, there is nothing but the absence of organelles and the biconjugate nature of the RBCs. These are the adaptations of RBCs for the transport of oxygen quickly and faster. Okay. Now what is the average lifespan of RBC in healthy individual? It is about 120 days. That is called the lifespan, 120 days. Now, after completing the life cycle, where are they destroyed? The place of destruction. The place of destruction is nothing but the spleen. The spleen is the place of destruction. That's why it's called a graveyard for RBC or cemetery for RBC. Graveyard or cemetery for RBC because it's a place of destruction. So after destruction, the iron components are released. The hemoglobin is converted into, that one, the bile pigments. The iron components are transported to the bone marrow for reuse. That's about actually the hemoglobin and its fate. Now, when you are reaching what is called high altitudes, I mentioned already you have developed mountain sickness. In order to overcome the mountain sickness is because of low partial pressure of gases and low partial pressure of oxygen in the mountains. To overcome this one, we have to increase the content of hemoglobin, thereby maximizing the acceptance of oxygen from the low partial pressure. What is the mechanism? Let's see. What is erythropoietin? What is its role? Erythropoietin is a hormone secreted by kidney in response to low oxygen. For example, when a person is reaching mountains, he develops acute mountain sickness because of low partial pressure of oxygen. To overcome this one, in order to increase the number of RBCs, the hormone that is erythropoietin is secreted when there is a low partial pressure of oxygen as in the mountains. What is its role? This hormone helps the differentiation of the stem cells of the bone marrow to RBCs. This process of formation of red blood cells is called what is known as erythropoiesis, particularly RBCs. Generally it is called hemopoiesis. The formation of RBCs is called erythropoiesis, or we can say hemopoiesis. Hemopoiesis is a general term used to develop or to form the blood cells commonly. Erythropoiesis refers to the formation of RBCs. Then, what is hematocrit or PCV? So we can just take the ratio of the volume of RBC, not number, the ratio of volume of RBC to the volume of the blood plasma. So that is being expressed as what is called hematocrit or packed cell volume. If you are taking a test tube and taking the blood and being actually allowed to centrifuge in a centrifuge, what is happening? The RBCs. This is the RBC which gets settled. Above which we have what is called the plasma, and also we have other cells in a low concentration. Now here we are taking the RBC as volume. So the ratio between the volume of RBC to the total volume of blood plasma is called hematocrit. But what is given in the book? The ratio of red blood cells to blood plasma, that is nothing but in terms of volume, not in terms of number. The ratio between the volume of blood plasma and the RBCs is expressed as hematocrit or packed cell volume, or simply PC. So after centrifuging the blood plasma or simply the blood, we are getting the RBCs get settled at the bottom in a known volume. And then we have the total volume of plasma. The ratio between the volume of RBC to the total volume of blood, that is called what is known as hematocrit or packed cell volume. Now with that I concluded about the RBCs. Now we have to go for white blood corpuscles or leukocytes. As a rule, you know that one, these WBCs are colorless, amoeboid and nucleate. and without hemoglobin and the pigments. Hence it is colorless in nature. If we are taking the total number of WBCs, it is least in number when compared to RBCs. RBCs are found in millions, but WBCs are found only 1,000. So approximately we have 6,000 to 8,000 WBCs in a known volume, namely 1 cubic millimeter of blood. So we have 6,000, 8,000 WBCs per cubic millimeter of blood. That is about the number, least number. The ratio I mentioned already, 1:700. For every WBC in that place there are about 700 RBCs, because RBCs are in millions, here they are found only thousands. So we have 6,000 to 8,000 WBCs per cubic millimeter of blood in an average healthy individual. Now we have to go for the different types of WBCs. Based on the presence or absence of granules in the cytoplasm, the WBCs are classified into two types. Those with the granules called granulocytes, and those without granules called agranulocytes. Okay. What are the types of WBCs? Based on the presence or absence of granules in the cytoplasm, the leukocytes are classified into two types. One, granulocytes, another one, agranulocytes. Now, granulocytes, those WBCs with the granules in the cytoplasm. Okay, there are the following types: neutrophils, eosinophils, and basophils. These cells are named based on the reaction with the dyes. Neutrophils react with, that is, neutral dyes. Then eosinophils react with eosin dyes or acidic dyes. Basophils react with the basic dyes, the coloring materials, and taking the color. The agranulocytes are classified into lymphocytes and monocytes. Now let's take the first one, granulocytes. So granulocytes are characterized by the presence of granules in the cytoplasm, hence the name granulocytes. Where are they formed? They are differentiated in the bone marrow. Their formation, they are differentiated in the bone marrow. That's about the differentiation. If we are taking the granulocytes, they are differentiated in the lymph glands and what we have actually the spleen. Okay, we'll talk about later. Now, the granulocytes are the following types: neutrophils, eosinophils, and basophils. We'll see one by one, a few points for each. The first type of granulocytes are neutrophils. They also have other names. It's an example of heterophils. Polymorphonuclear cells. Polymorphonuclear cells because the cells with 3 to 4 lobed nucleus. The nucleus is multi-lobed, hence the name polymorphonuclear cells. The lobes of the nucleus are connected with the delicate threads. So these are the largest in number when compared to other types of WBCs. So they constitute about 60 to 65% of the total number of WBCs. What is their role? They are phagocytic in function. They are also called as wandering phagocytes. And appear in large numbers when there is an infection in and around the infected tissues. So we can find out whether the person is infected with any bacterium or not by counting the number of neutrophils. During infection, the number of neutrophils increases in and out what we have in and around the infected tissues. Now this is what we call the neutrophil with the multi-lobed nucleus with the granules in the cytoplasm. Now the next one is eosinophils. These cells have distinctly bilobed nucleus. You see that one bilobed nucleus with the granules in the cytoplasm. And the lobes are joined by, we call thin strands. There is a thin strand connecting the two lobes. Now they are not doing any phagocytic function. Hence we can say they are non-phagocytic. They constitute about 2 to 3% of the total number of what we have WBCs. Then what is their role? Their number increases normally during certain parasitic infections or during allergic reactions. We have one type of allergy caused due to dust that results in increased number of what is called eosinophils. That is called eosinophilia. Eosinophilia is due to increased number of eosinophils, a kind of allergic disease. Okay, that's about what we have eosinophils. Now we have to go for the next one, the basophils. The third type of granulocytes, basophils. They are less numerous than any other type of WBCs because they constitute only 0.5% to 1% of the total number of WBCs, least in number. Now the cytoplasmic granules are large size but fewer than what we have eosinophils. You see, the granules are larger but less in number when compared to the eosinophils. Now the nucleus is large sized and is constricted into several lobes, but the lobes are not joined by what we have, that is delicate threads. Then, what is the role? They secrete very important substances taking part in inflammatory response or inflammatory reactions. What are the substances secreted? They secrete what is called heparin, an anticoagulant, which prevents the coagulation of blood inside the blood vessels. Histamine, the one which is taking part in allergic reactions. Serotonin, another substance involved as chemical alarm signals. So these are all the substances. Histamine and serotonin related to allergic reactions, and heparin is related to prevent the coagulation of blood, anticoagulant. So, because of releasing these substances, their main function is related to inflammatory reactions. We have inflammatory response. That response is created mainly by the histamine along with other alarming signals, chemical alarm signals. They are histamine, serotonin, etc. Prostaglandins also. So this is about the structure. We have a bilobed nucleus, more or less it is sigmoid shape or S shape. Okay, that's about the basophils. Now we have to go for the second type of WBCs, A granulocytes. These cells are characterized by the absence of, that is, granules in the cytoplasm. And where are they differentiated? They are differentiated in the lymph glands and the spleen. Now what are the types of A granulocytes? We have to see. The second type of WBCs are A granulocytes. As the name implies, they are characterized by the absence of granules in the cytoplasm. Where are they differentiated? They are differentiated in the lymph glands and spleen. Now what are the types of A granulocytes? There are two types of A granulocytes. Number one, lymphocytes, and number two, monocytes. Now what are lymphocytes? What is their number? They constitute 28% of the total number of WBCs. They have a longer lifespan, even about 300 days. That's not given in the book. They have a large round nucleus and a small amount of cytoplasm. The nucleus is large and the cytoplasm is normally little amount. So the proportion of nucleus and cytoplasm, if you are comparing the ratio, the nucleus is larger, but the amount of cytoplasm is less. Now what are the types of lymphocytes? There are two types. B lymphocytes produced in the bone marrow. T lymphocytes, otherwise called as thymocytes. We'll be studying later in immunology. Bone marrow lymphocytes and thymocytes are thymus lymphocytes. And both B and T cells are involved in the immune response of the body, the defense mechanism of the body. Now B cells produce antibodies to neutralize the foreign substances, to neutralize the harmful effects of foreign substances. What about the T cells? The T cells are involved in cell-mediated immunity. Antibodies, otherwise called humoral immunity. Then we have the T cells that are involved in cell-mediated immunity. We'll discuss more and more in immunology in 12th standard. So B cells produce antibodies to neutralize the harmful effects of foreign substances, and T cells are involved in cell-mediated immunity. That means the cells are involved in providing immunity. That's about what we have B and T cells. The B lymphocytes at the time of production of antibodies, it is called plasma cells. So it changes into plasma cells at the time of production of antibodies. Okay, let's see now the next one, the monocytes. As I mentioned earlier, B lymphocytes produce antibodies and T cells are involved in cell-mediated immunity. Now what about the monocytes? Now normally if we are taking the lymphocytes at the time of production of antibodies, they are called plasma cell. So some B lymphocytes become plasma cell. That is nothing but antibody producing cell. Now the monocytes. There is also another name for this one, macrophages. These cells are mainly phagocytic in nature, similar to the mast cells of the connective tissue. In the connective tissue, there is one type of cell, what is called the mast cells, responsible for the release of histamine, like what we got the B cells. I made a mistake. Basophils, the mast cells present in the connective tissue, and that function is also similar to the macrophages. The special feature of this monocyte, kidney-shaped nucleus or reniform nucleus. They constitute about 1 to 3% of the total number of Wbcs. Now the macrophages have different names in different places. In the central nervous system, it is called microglial cells, phagocytic in function. Now in the liver sinusoids, the cavities, the spaces, they are called Kupffer cells, concerned with the phagocytic function, swallowing and disgrading what we have, that is the Wbcs. Then in the pulmonary region, they are called alveolar macrophages. So different names in different places. At the time of just actually wandering here and there in order to capture the bacteria and other dishes. Now some monocytes become what's called the macrophages. So it is nothing but the modified functional monocyte. And similarly, the plasma cell is nothing but a modified functional lymphocyte. That is particularly what we call the B lymphocyte. At the time of production of antibodies, it is called plasma cell. Similarly, at the time of function, the monocytes become the macrophages, having, that is, bacteriolytic enzymes, some what is called radicals, in order to just phagocytose the bacteria. That's about what we have the granulocytes. We have completed the Wbcs. And one more thing, just in the blood formed elements, the what are called the thrombocytes, otherwise called the platelets. A few lines only. I will complete the next page. The next one in composition of blood, thrombocytes or platelets. Normally they are produced from a large sized nucleated cells. A cell with a large nucleus, we can see. The cell is not large, the nucleus is large. And such cells are called megakaryocytes. A specialized type of cell found in bone marrow. These cells undergo disintegration to form the platelets. As a result, these cells, normally we cannot say these cells, the platelets are without nuclei. So they are lacking nuclei. What is the number? There are about 1,50,000 to 3 lakh platelets in 1 cubic millimeter of blood. What is the role? They secrete certain substances which are taking part in the clotting or coagulation of blood. When there is a reduction in number of platelets, that result in what's called the loss of blood from the body, leading to some disorders that can lead to loss of blood from the body. So reduction in platelet number can lead to some disorders, what we call the clotting disorders, that result in loss of blood from the body, because these cells are responsible for the secretion of certain factors which are taking part in the clotting or coagulation of blood. So with that I concluded about the composition of blood. We have to go for the next aspect of circulatory system. I will do it in the next class. Thank you.